Okay, let me put this PowerPoint on here real quick. All right, excellent. Well, I was told last night something I've never been told before in my entire life. It's that I was soft-spoken. Soft-spoken. I didn't know whether to take that as a compliment or not. So they said, "Hey, you're awful soft-spoken. You need to speak up so everybody can hear you." Is everybody be able to hear me now? Can you hear me over there? That hearing aids, or is it just? It's better. Good, good. You know, I was going to come up and say that I'm feeling something really, really heavy in my stomach right after breakfast here, and it had to be a biscuit, but it wasn't a biscuit. Those were delicious, and I will mention that tonight. I know all the guys were giving you a hard time saying we're going to thank your wife for everything, but thank you for the effort you put in. What was that?
She had a big part of it. I just didn't use her recipe. She was a little tipped at that.
All right.
She liked it.
So is that like, so when you're saying she had a big part of it, does that mean she did 80%, 90%?
Team effort.
Team effort. Okay, no percentage, just a team effort. And congratulations on your 52 years of marriage. What a wonderful. Pardon me?
Steve supervised.
I can do that too.
Okay, good, good. Well, praise the Lord. Now, some of you don't know that I actually grew up in Cheyenne. So you don't get very many speakers that grew up in Wyoming. So I grew up in Cheyenne. My dad was in the Air Force, and he was stationed at the Air Force base that's down there in Cheyenne from the time I was in first grade and until I graduated from high school. And during that time, so he stayed at one base pretty much the entire time of his military career. He had a little stint in Florida for about 3 years during the space program. He was the base down in Cheyenne is a missile base, and they don't have any airplanes down there. It's all intercontinental ballistic missiles. That's what they run. And he was a surveyor. He retired as a master sergeant, and he did surveying for targeting for the missiles. And then during the space program, they needed his services for the space program. And then we went right back to Cheyenne, and I stayed there till I graduated. We would play Gillette and football, so I'd be either up here or down there. I never knew when I was in high school how they got the name the Gillette Camels on that. It's like, where did the Camels come from? It was, we were the Cheyenne Central Indians. That made sense, but the Camels, I didn't even know anything about that. And then when I graduated from high school, instead of going to the University of Wyoming, I transferred and went to South Dakota School of Mines and Technology up in the Black Hills. And that's when I realized, the northern part of the Wyoming, South Dakota area is much nicer than the windy southern part around Cheyenne. And I ended up falling in love with the Black Hills, this whole area here, and ended up buying property. And I had some property in South Dakota, and I bought some just north of Newcastle, and then I own a piece right up here near Devil's Tower. So I'm always over here a lot, and in many ways, this is a vacation. So I know you say, "Oh, thank you for coming up here." I say, "Thank you for inviting me." It's a wonderful break from all of the masses, and I mean masses of people down in the Dallas-Fort Worth area. So it's really, really nice to be up here. Pastor mentioned, he asked if I could say something about some of the stuff that's happening at ICR. So very, very quickly, since I took over as president, we have made probably 3 or 4 major changes. The first and foremost is the one we're going to talk about this morning, which is how ICR's cutting edge research, I'll go ahead and just put the next slide up there, confirms engineered adaptability. And the major changes I covered in many ways last night in our new mission statement. So ICR was initially started as a research institute. And for about the first 25, 30 years, we were very, very heavy into research. We were the only creation ministry, really the only game in town. So anybody who was teaching creation science was using things that were developed by the Institute for Creation Research. And in many ways, we set the arguments. Here's the argument against uniformitarianism. Here's the arguments against evolution and other things. And we also tried to build some models. And then we kind of, and I would almost say drifted a little bit away from our core ministry, which was research, scientific research. And we were more interested in presenting the information in an enticing way to get people to come to our ministry. And so we ended up building a very, very nice facility, a ICR Discovery Center. It's like a big creation museum that's down in the Dallas area. But you only have so many resources, and so in order to build the museum, the scientific research kind of fell by the wayside a bit. Never gone, but de-emphasized. So since I become president, research is back on the top pedestal. That's what we're here for. That's what we exist for. That's what we give people in the church, is answers to questions which are threatening pastors, Christians, everybody, everywhere. Second, even though the whole world was going into the digital realm, everything was going online and people were looking online and things like that, we didn't really have an online presence anywhere. We had a few programs, we had a few little spots that we did, but we didn't have an online presence at all. So we started from scratch, brand new, new digital media department, which we have a major online presence now. Multiple podcasts, different types, different styles, hopefully reaching different groups of audiences, everything from younger to older people, and those who are heavily into the science and those who really aren't, just not really interested in science that much, but we still want to get a message to them. So across the board in every way, we have a brand new digital media department. It's exploded in size. We went from nobody to having 7 people in the department plus 2 hires. We basically are full-time people, work for ICR, but they live overseas, who do animation for us. And that has really boosted our presence online, which is very, very important in those areas. Third, you'll notice there are a lot more books on the table and a lot newer books. And they're not all related to heavy scientific content, which they used to. So this is another area to reach more people with the creation message. So we went from being ICR Press to ICR Publishing Group, where we have 6 different imprints where we are publishing materials from everything from our heavy scientific materials all the way down to children's materials. And on this end of the table, you'll see there's a lot of children's books, children's materials. There's some little biographies there. I saw one was on George Washington Carver. We have one on Kepler. We're having another one come out on Newton. So we're trying to publish information all the way across the board so that now a lot of our followers who have been following us for 50 years, they're in their 60s and 70s and they have grandchildren and they're very concerned that they get information. We have something we can put into their hand along those lines. And then fourth, and this is the other major area, and this is what we'll be talking about today, we are moving away from our traditional explanation, which I will get into this talk, and I just added a few slides based on a conversation I had this morning at breakfast. Our traditional explanation, which you'll see, was just a lighter version of evolution towards a new engineered biology. It's an organism-focused, not environment-focused, an organism-focused, engineering-based explanation to explain all of your biological functions. So you'll see how this works, and I'll explain it to you. So we are basically going with the assumption that organisms look highly engineered because they are highly engineered, and therefore we are going to use engineering-based explanations to explain all of those things. So before I start, you'll notice I'm drinking a lot. And you probably noticed last night I had to just drink and drink and drink water. This isn't water. It doesn't foam like that normally. This really is Mountain Dew, but I didn't want to, since it's being filmed, I didn't want to, advertise for Mountain Dew on those kinds of things. That's because in June, I had to have a horrible surgery. I mean, one that I wouldn't wish on anybody. And on both sides of your cheeks, you have big glands that are salivary glands. They're called your parotid glands. Well, my right one got a tumor in it. And so I had to have the entire gland removed in a surgery. So they make a big incision and lift half your face off, get in there and remove this gland. A nerve goes right through the gland that controls your facial expression and your muscles. So they have to dissect the gland off of the nerve without nicking the nerve or stretching the nerve or damaging the nerve and giving you a facial paralysis on there. And then, stitch your face back on. And so I had that surgery in June, but I lost half of my salivary production. So you're talking and talking and talking. And there's one thing that you need to talk, and that you need saliva. So that's why I take these drinks every now and then. Even though Mountain Dew is somewhat counterproductive to that, it's good for in the morning. So that's why I drink it here. All right. How cutting edge research? This is ICR's research. Can you hear okay? All right. Are you moving up because I'm not talking loud enough? It's better now? Is everybody else, can everybody else hear me okay? And I just want to know if it's me or not. And that you're all okay? Good? Louder? All right. All right. First thing, I have the sign-up sheet in the back. I suspect most of you have already signed up for our magazine. I have an article in almost every single issue of Acts and Facts related to the engineered approach to biology. There's always something new that I'm able to write up and discuss and bring about. So if you haven't signed up for this and you're interested in an engineering-based approach, and most guys are, to biology, this will keep you up to speed with our latest findings and other things like that. So we want to, as I mentioned last night, not just talk about God. We want to expose the world to Jesus Christ. So here is the link. When it comes to evangelism, you talk about Jesus Christ and people say, I don't believe any of the things that are written about him. In fact, someone even mentioned that this morning to me. I don't believe these things about Christ. Well, why don't you believe things about Christ? Well, I don't believe the Bible. What don't you believe about the Bible? I don't believe the Bible's the Word of God. Well, why don't you believe the Bible's the Word of God? Because it's full of errors. It's full of scientific errors. And so that is the link. They don't believe the Bible because they believe the Bible is wrong, and because they won't believe the Bible, they don't believe what the Bible says about Jesus Christ. So that's why our ministry is so vital, not just to truth, but even to evangelism. People have to believe the Bible before they'll ever believe a man who healed the sick, raised the dead, rose from the dead, and died on a cross as the substitute for all of their sins. So ICR research in many ways is so important to help people understand that they can believe the Bible. And one of our major ministries and missions, as I mentioned last night, is to change Christians' worldview. Not just Christians, but everybody's worldview by constructing an organism-focused theory of biological design. That it will highlight Jesus as the creator. Now there's a key word, organism-focused. As you will see as I go through this men's breakfast here real quick, evolutionary theory is not focused on creatures. It's focused on the environment. It's focused on the environment as the shaper and molder of creatures. From their worldview, creatures are the way they are in all of their great diversity because the environment has slowly molded and shaped them over time to fit the various environments that they live in, which has led to the diversity of creatures around the world. But far more important for their thinking, it's led to the reason why creatures look so highly engineered. Why do birds have such incredible flying features? Why do they put the best engineered planes that humans have made to shame? Well, it's because nature has slowly shaped them over millennia, literally millions of years, one little change at a time. With every environmental change, it has shaped these creatures so that they fit these environments, and that's why birds look so highly engineered. So evolutionary theory at its core is not to explain the evolution or diversity of life. Evolutionary theory at its core, maybe this is better, it is fundamentally an anti-designer, anti-engineered explanation for life. That is what Darwin is after, because everybody who has ever lived looks at creatures and says there must be a God. They're not talking about Jesus, they don't know, but they're saying there must be a God. Things don't make themselves, these things look like they're made. How in the world did that happen? So ICR is taking on one of the major icons of evolution, and that is these fish that are absolutely blind and low in pigmentation that live in caves. And I'll give you their explanation for it in the long run. So this new engineered-based approach is not just an answer to evolutionary assertions. And it is not just identifying a step in a biological process. And that's what so much research is after. It's just like, well, how did this work? What's the next step? What ICR research really is at its core, it is a test of really important assumptions. It's the test of the assumptions of evolutionary theory. Evolutionary theory will say organisms are shaped by their environment. Evolutionary theory will say change, genetic change, happens through random genetic mutations, random mistakes. Evolutionary theory will say that systems and organisms get broken down by these random genetic mistakes. So that's what they claim in their theory. We can put that to the test. And we can offer an alternative explanation that says, no, organisms actually continuously track environmental change. So what has been the conventional explanation for cavefish characteristics? Well, let me read to you the placard in a museum, a creationist museum, that explains these cavefish. This is what this creationist museum says. And this is a direct quote right from it. As genetic information is copied and passed on generation after generation, occasionally there are copying mistakes known as mutations. So as your cells replicate, they copy their DNA so that it can split into two cells. In the process of copying that, a mistake might happen. Or maybe you get bombarded with a cosmic ray and it changes one of your DNA nucleotides. Mistake. Or maybe you get exposed to a chemical and it's called a teratogen and it causes a mistake. Somehow a mistake happens, and those mistakes are mutations. Mutations have been observed to destroy, damage, or corrupt genetic information. That is true. Or to be neutral. In other words, they're not going to kill you, and even if you pass them on to your children, they're not going to kill your children. So that means they're neutral. They're not helping you, but they're not killing you. But have never been observed to add new information. This is true even of so-called beneficial mutations that may be advantageous to the surviving organism in some circumstances. This went on to say, when a mutation occurs in a light environment that causes animals' offsprings not to have eyes, so they're talking about the blind fish, it's an enormous disadvantage. So natural selection eliminates this flaw. When the eyeless defect occurs here, that is in a cave, it does not cause a disadvantage because not having eyes in the dark is not hurting you, is what they would say. So it is not eliminated. In fact, it gives advantages. Those with eyes can crash into things, injuring the eyes, and can get diseases of the eyes, possibly leading to death. Eventually, selective pressure ensures that all are eyeless. These ghostly fish are prime examples of how mutations in natural selection lead to a reduction of functioning systems. These adaptations are no evidence at all for the belief that complexity has arisen by such processes. They only show how information can be lost in a fallen world. That explanation was in the Institute for Creation Research, original museum. That is how we explained blind cavefish. So if you summed up our explanation about the blind cavefish, we claimed that surface-dwelling fish that lived on the surface in streams that eventually found their way into caves, gradually morphed into a cave-dwelling form, which was due to random genetic mutations, which led to broken systems. That's why you don't have eyes. It's because the mechanism to develop eyes was broken, which was due to, which was sifted through a purposeless trial and error process of death and survival, i.e. natural selection, that ended up resulting in a loss of information that produced depigmentation and blindness, and then a selection pressures eventually ensured that all are eyeless. Now I pause here for you just to ponder what we were telling everybody. Think about it. What is the difference between our explanation, and what an evolutionist would explain? There's no difference. Evolutionists would say they were sighted and they went to the cave. Mutations cause broken systems. A struggle to survive led to the ones with smaller and smaller and eventually no eyes over a long period of time. And all we would add is, well, this shows a loss of information, not a gain of information. May I suggest to you why ICR and others have made far more progress in the geological realm by offering an alternative explanation to slow periods, by offering an alternative explanation of the flood laying down layers, was far more formidable because it offered a completely different explanation. And this explanation in biology has been far less formidable over the last 50 years, is because it's just a lighter version of evolutionary teaching. It's not an alternative explanation at all. It just says it's real. What Darwin claims is real, what evolutionists say is real, but it just can't do everything they claim. Are you following me? We're saying your evolutionary process is real, but limited. What we should have been saying for 50 years is it's not real. It's not true. By conceding that the entire process was workable and real, we were conceding way too much, and we were playing Darwin's game on Darwin's field by Darwin's rules. Now, and then we would throw in this little caveat here. All it shows is how to break something and not make something. In other words, you started out with eyes that God made, and then they got broken over time, and there we were lost. So you're not explaining the evolution of eyes, you're explaining the loss of eyes. Now this is where it gets a little complicated. We should have been saying this whole explanation is wrong. It's totally wrong. So let me just appeal to you without even giving my explanation. Knowing what you know about the Lord Jesus in terms of his genius and his power and his wisdom, knowing that. Number two, knowing what you know that the Lord gave animals the command way at the beginning of creation to be fruitful and multiply and fill the earth. Knowing that you know the Lord gave this command to the creatures. When God gave the command to the creatures to be fruitful, multiply, and fill the earth, would that have included caves? I'll get a drink while you ponder that. Would that have included caves? Yes. It includes caves. It includes Arctic environments. It includes deserts. It includes the bottom of the ocean. He said, be fruitful, multiply, and fill. You fill all of these different environments. And when these creatures fill these environments and they're living in a cave, they're living at the bottom of the ocean, they're living in extreme heat or extreme cold or whatever it is, you, as a human being, you say this, I know you do. You say, wow, those creatures are really cool. Those creatures can really do incredible things. Look at where they're living. And when you say, wow, look at those incredible creatures, what you're really saying is, wow, look at the one who made these incredible creatures to do these incredible things. May I suggest to you, incredible stuff is what brings the most glory. Does that make sense? If creatures just did simple little things, what glory is that? Even with human beings. If you want to know how good human beings are, you don't build a chair, or as millennials do today, nail a couple pallets together and call it their chair, or something like that. No. If you want to know how good human beings are, you put a man on the moon. You do something that is really hard. You do something that's a huge challenge. And when you accomplish that challenge, everybody says, wow, we just put a man on the moon and got him back alive. So doing incredible things is what really reveals how glorious the Lord Jesus is. Does that make sense? So, the Lord made it so fish with eyes and pigmentation and all kinds of other traits could somehow, we don't know how they really get into the caves. Do they swim in there on purpose? Do they get washed in? Whatever it is, they find their way into a cave. Now they're living in a cave in absolute darkness with minimal food, with changes in the water chemistry, low oxygen, high carbon dioxide, very acidic water, on and on and on. The differences in the cave between that and a stream are remarkable. How in the world do they do that? So here's my question to you. Knowing that of the things about the Lord, knowing that he made them live in the cave, which is in truly challenging conditions, does it make any sense to you at all that he would do it by breaking genes, breaking systems, and having broken down stuff that just happened to possibly maybe solve some problems and work? I'm asking you guys. No. Why? Because no human engineer in their right mind would ever do it that way. No medical doctor would ever experiment on you, one broken trial and error, maybe get it right, hopefully get it right, one death after another. Oh, finally I got it right on you. Nobody does it like that. Nobody with a real brain. So when we were ascribing this mechanism to the Lord Jesus, not only was it conceding evolutionary theory, may I suggest to you, it was fundamentally insulting. Fundamentally insulting to the Lord. Degrading to the Lord. So, how would a real engineer do it? Well, I can tell you this. When we made a rocket and a system to put men on the moon, we built into the systems before they ever got to the moon and landed on the moon the qualities in those systems which would enable them to survive on the moon, live on the moon, function on the moon. And therefore, probably from the very beginning of creation, the Lord built into these fish the ability to live in a cave. Before they ever got to the cave, it was already there in them. Does that make sense? That makes sense. So he built into them how to live in the cave. And now the challenge and the joy of human beings is to discover that. He gave us that. That's a gift. When we can do research and we can discover things, it's a gift. It is a real gift. Every day you do research and you find something, it's like unwrapping a Christmas present. Oh, look inside. Look what we found. There's this joy, there's this excitement, there's this anticipation that you get when you discover something and unload them. So, continuous environmental tracking, which says organisms were engineered up front to continuously track their environmental change and adjust themselves to these different environments, is the first really anti-Darwinian explanation for biological adaptation. Why is it anti-Darwinian? Because Darwinism, Darwin's explanation, was fundamentally anti-design. And so since ours is design-based, it's anti-Darwinian because it's moving us back to an engineered or design base of biology. And it's the first model offering creationists and intelligent design advocates that is a thoroughly engineering-based explanation. It's completely organism-focused. What do I mean? I mean, if we send a man to the moon and you want to understand all the technology that goes into doing that, you don't study the moon, you study the rocket, you study the lunar modules, you study the life support systems, you study everything that was built into those, you focus on the thing that was engineered. It uses purposeful, non-random mechanisms, and it rejects all mystical selectionist reasoning. Why is selectionism mystical? Because in their view, when they see organisms with these traits and the environment changes, and now they express traits which fit the new environment, they say the environment selected for those traits. They say the environment favored them. They say the environment acted. In other words, they put all of these action verbs to the environment to select, to favor, and to act, which is nothing but mysticism, because the environment can't select. The environment can't favor. The environment can't act on. It's not alive. It doesn't have a brain. It can't do any of those things. So we look at the things that are actually changing. So here's a good summary slide. What is continuous environmental tracking? Well, as I mentioned, it's engineering-based. On the left-hand side of the screen is a car. This is actually an older picture of one. This is well over a decade old. Obviously, it's made by General Motors and also sponsored by Caterpillar. You'll see all those antennas and everything sticking out all over the car. This is a great picture because it actually shows the sensors. This car is unmanned. It's completely autonomous. If it could carry enough fuel, it could drive from New York to Los Angeles. It could drive around traffic jams. It could drive around bad weather. It could drive around all kinds of hazards, all by itself, without even a human being sending it messages, telling it where to go. It is totally autonomous. It collects its information, it processes on its own computers, and it responds accordingly. In other words, there's not a link from the audiovisual guy in the back changing it. It does it all by itself internally. Now, is that pretty impressive? That's pretty impressive. You're just like that. You're like this. You have sensors. You can go around things. Your body, right now, your body is digesting those biscuits. You're wondering, wow, maybe that's a big hard thing or not. But what do I mean? It's breaking them down. It's breaking down the sugars. And the sugars are now being processed for energy. And now they're supplying your brain. In fact, most of the biscuits are sugars. There's these long chains of carbohydrates. And it's breaking it down, pulling it apart bit by bit, reabsorbing it, transferring it through your blood, and it goes into a powerhouse which makes a molecule called ATP, which then powers the rest of your body. And you're not having to think about it. You're not having to work on it. Your body just does it. Boom, 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 boom. Completely self-contained and self-controlled. Well, that's what's happening here. It has sensors. It's got computers. It's got output responses, and it can respond appropriately. Well, you do the same thing. And then you'll see there's corresponding system elements. So on the right-hand side of the screen, can everybody read that headline? Guinea pigs beat climate change by tweaking their own DNA. Now, this is kind of cool. Right now, your DNA is being changed. It's being changed in real time. You think, well, my DNA is my DNA. No. Your DNA, just like other things, can be modified and adjusted. And in this case, they did an experiment on guinea pigs because everybody's wondering how they're going to respond to climate change. So they took 5 male guinea pigs, and they have them in their cages, and they put a heating pad in the bottom of the cage. And they cranked the heat up on the heating pad to mimic global warming. How are the guinea pigs going to work when they're being cooked? Well, if you know anything about male guinea pig anatomy, what's sitting on the heating pad? Right. They're right down there getting roasted. And they cooked them for 70 some days. Why for 70 days? Because it takes that long for a stem cell to divide into an immature sperm cell to become a mature sperm cell in order to fertilize a female guinea pig egg. It takes 70 days to go through that cycle. So they run through 70 days to go from a stem cell to a mature sperm cell to fertilize the female guinea pig eggs. By the way, the females were never put in hot cages. They just lived normal lives. She had pups. And then they can check for genetic and epigenetic changes on the pups. And in that one generation of cooking dad, these pups had at least 18 different changes to their genes. And the genes that were changed were specifically related to living in a hot environment better. Not only did the pups have those changes, but dad had already begun to express those same changes. Not over millions of years, not through random genetic changes. It is a built-in mechanism, which, as the headline says, enabled them to tweak their own DNA to live in a climate that's hot. Now, is that cool or what? That's pretty cool. That's pretty cool there to do that. And it does it the same way the car does it. It has temperature sensors. It has logic. It has a way to change the DNA. It has a way to put little markers on the DNA saying crank this gene up or turn this gene down. That's what's happening. And that is really, really remarkable. That's really cool there. So continuous environmental tracking is this, it focuses on the organisms and how they self-adjust inside to their changes in their environment. And when we look at organisms as they continuously track environmental changes, the characteristics that come up to describe their adaptations are not slow and random. These are the words in the scientific papers: regulated. Adaptation is regulated. In fact, most papers say highly regulated. Not slow, but rapid, reversible, repeatable, and with responses, like with the guinea pigs, which are so targeted to the change that they're even predictable. Regulated, rapid, repeatable, reversible, and targeted. Those are the descriptors of when organisms rapidly are able to change. And it's the organisms which are the agents in control. So here is the main question. How in the world do we explain these changes? So here's the fish up on the screen. The surface fish is the one up at the top. It's got eyes, it's got pigmentation. The cave fish is below. You can see no eyes, no little pigmentation. They're pink to whitish in color. They have many other changes. Their jaw has changed, their head has changed, their skull has changed. Their red blood cells have changed. The metabolism has changed. When those look at which one of those fish looks fatter, the cave fish or the surface fish? The cave fish. That's because the cave fish their metabolism is changed. That when they eat food, it almost goes immediately to fat. It gets stored as fat. Why? Because fat is how you store your energy, and in a cave, your next meal may be a long ways away. So in medicine, we used to say, you feed the fat, and then the fat will feed you on that. Feed the fat, and the fat will feed you. And fat is the way that the Lord designed us to store energy. When you have an abundance, you build up the fat. And then all of us have gone through this. You go through periods where you don't get as much to eat, or you work like a dog, and pretty soon the fat goes down. It's just that way. These fish are doing the same thing. Their metabolism has changed. Same question. Here's just a different picture of them. Surface fish, cave fish. You can see the differences. Pigmentation, color, eyes, and those things. By the way, even though these fish, they're very different, they're the same species. And the surface fish can mate with the blind fish. They mate together, and their offspring have eyes. They're born with eyes. So they can mate together. By the way, this is kind of cool. You can easily see how two surface fish, a male and a female, could get together to mate. But how do you do it in a cave when it's dark and you can't find each other? Well, they do. In fact, we have filmed these cave fish mating, and it's utterly remarkable. Totally blind fish. They find each other, the male and the female. They go through the same kind of rituals that surface fish do. And when she's ready to release those eggs, and nobody knows how the male knows this, these two fish, by using other sensors, not their eyes, other sensors along the lateral line is that right there. How many of you caught trout that have like a pink stripe down their lateral line? That's not just there for coloration. It is chock full of sensors. Sensors that can detect in the water things that are close to them. So it is a sensor that's able to detect when something is close to you without even having to touch it. Now that is really cool. It's like what you would find on a submarine. So these two fish, using their lateral line and these other sensors, they will swim in a choreographed motion very rapidly, and everything happens almost within 1 second. They swim, and if you catch this on high-speed photography, you'll see that the female fish will swim inverted for just a split second. Just a split second, she's swimming upright, and then she flips inverted, and at that very split second, the male will flip his tail up and touch. She will release her eggs into the water. Eggs are not activated to be fertilized until they touch the water. When they touch the water, then they can be fertilized. Sperm is not activated to be able to fertilize an egg until it touches the water. So at just the moment she releases the eggs and they are sprayed into the water, the male will release the sperm, spraying it in, and it finds those eggs and fertilizes them. And then in split second, she swims back upright again, and they swim off, never talking to each other. Just boom, all within a second. Highly choreographed, not using their eyes. Now that is a really cool engineered mechanism as well. Well, how in the world did all of this come about? I'm going to just read to you just snippets of some quotes from the evolutionary literature. This was from, what year was this? 2004, 2002, so about 20 years ago. Early estimates placed the divergence of blind Astyanax, that's the name of the cavefish, morphotypes from sighted surface fish at about 3.1 to 8.1 million years ago. So it took that many years to go from a sighted fish to a blind fish. 3 million to 8 million years ago, according to this evolutionary explanation. And then, by 2019, a decade later, William Jeffrey of the University of Maryland reported a much younger age saying, quote, surface fish and cavefish split from a common ancestor very recently within the past million years or less. So it went from being about 3 to 8 million years ago to now being about 1 million years ago. And then a newer paper, 2020, says they split from each other about 200,000 years ago. So it's gone from 8 to 3 to 1 to 200,000 million years ago. And then another paper, about the same time, has the population splitting less than 20,000 years ago. And another paper recently says, well, maybe they've just split very, very recently from each other. So the evolutionary trend in terms of the time of this divergence is down, 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 down. It is totally plummeting, totally plummeting. Of course, as creationists, we would say they just diverged from each other within the last few thousand years, sometime after the flood. Now, in terms of mechanism, there is a big difference. As a creationist with an engineered-based explanation, I would say these fish were engineered with the ability to fit caves right from the very beginning. I would say they have built-in mechanisms which are functioning that can be dialed up or they can be dialed down as necessary. I would say they can track environmental changes, which means they can have pigmentation and no pigmentation. Evolutionists have a completely different explanation. From what I would say creatures can do this over and over and over again. But evolutionists have a different explanation. So this is their standard explanation. And all you have to do is just read the first sentence. Loss of function mutations in a gene which is called OCA2. And there's the long name, oculocutaneous albinism. In other words, you lose your eyes, you also lose your pigmentation. Has been characterized in 3 independently evolved populations of this fish. And while the effective gene is the same in all these cases, for reasons currently unknown, OCA2 seems to be, here's a personification, evolution's favorite for pigmentate regression in cave populations of these species. Let me translate. What they say is this: there's a change in a gene, and every population where we look, the same gene is changed in the same way for reasons we don't understand. I would say there's reasons why the same gene is changed in the same way. It's not through random processes. It's because it's being purposefully changed. Purposefully changed. It's not loss of function. Here's another paper. It says mutations have occurred in both Pacquion, that's one population, these fish are found in Mexico, and Molino OCA2 genes. Molino is another population in Mexico. But they are caused by different deletions in the protein coding sequences. Thus, the OCA2 gene is repeatedly targeted for mutation in cavefish, resulting in independent origins of albinism. That's really important. It says, when we go and we look at this population of cavefish, which are completely separated from this population of cavefish, which are completely separated from another population of cavefish, in other words, they're not mixing with each other, every population has independently arrived at the same genetic change. Now, how do you explain that? They would say, lucky chance, lucky chance, they all arrived by luck through random trial and error to arrive at the exact same changes. Does that make sense? Or is it more plausible that there's a mechanism which is changing it in this independent population and this independent population and this independent population? That makes more sense. That makes more sense at all. And then this, I'll just skip over this. It's another thing saying the same thing. We find the same genetic changes in independent cave populations. So evolutionary biologists assume that the observed genetic changes are random. Now, why do I say it's an assumption? Well, because there's no test to tell whether these changes were done on purpose or whether they were done randomly. I would suggest when you find the same thing in independent populations happening, you should be leaning against random, and you should be thinking purposefully on these things, on that. They think that these are broken genes which result in broken genetic pathways and a loss of function. And I would suggest just the opposite, that these are changes which can be dialed up and dialed down. So here's our experiment. This is a surface fish. We caught this right from a stream outside a cave down near San Antonio, Texas. In fact, we were able to capture a whole population of these fish. And they're still alive. We captured them 3 years ago, and they're thriving in our custody. So this is what a surface fish looks like. We're focusing on pigmentation. The eyes are going to take a lot of research to crack. So we wanted to focus on pigmentation. So this is what a surface fish looks like in a stream, exposed to sunlight in surface fish conditions. Here's a cave fish. This is a female right here. We purchased her from an independent farm where that raise these cave fish. Now we're able to breed our own and we don't have to purchase them. But at the time we purchased these fish, and here's what one of the cave fish looks like. We took this female cave fish and we re-exposed her to simulated surface conditions with sunlight at the same frequencies, at the same wavelength, and the same intensity as that fish would be exposed in a stream, under the assumption that pigmentation is not broken, it's just been dialed down. And if it can be dialed down, it can be dialed back up. So here's a picture of the same fish. Here she is, and that's what she looks like in 32 days. Not 32 years, 32 days. Boom, boom. That's pretty impressive. Pretty impressive. So my question to you is, was her pathway to make pigmentation broken? No, it wasn't broken. If it was broken, you could expose her to all the sunlight you want and she would stay hypopigmented. If it's something that can be dialed up and dialed down, then re-exposing them, boom, you get the pigmentation back. By the way, here's a male. Male was the same thing. He was kind of pink, and we put the male right in the same tank with the female, and he grew just as pigmented as she did. And then we mate those fish, and their offspring are born, and guess what they're born with? Hypopigmented or pigmented? They come out born and they are pigmented. Pigmented in one generation. So we are able to put assumptions to the test. Is this a broken system or is this a regulated system? Here's another experiment. We did it under more controlled conditions. There's the surface fish, and there is a female fish, and then we exposed her to intense sunlight, and there she is 60 days later. She's even more pigmented than the surface fish. More pigmented. So the systems are not broken due to random genetic changes. They can be upregulated and downregulated. And there's a comparison before and after. This is the same fish. Same fish. There's the male and female, both in the pigmented form. Then We took a surface fish and we exposed her to simulated cave water conditions where a lot of the electrolytes have been depleted from the cave water. And this is the same fish, this is a picture, going from a pigmented state to a hypopigmented state. Pigmented, hypopigmented, lost the pigmentation, and the loss of the pigmentation from this, from this picture right here to this picture was 16 days. 16 days. That's quite remarkable to go from this to that in 16 days. What a major change. And then we were able to document this pigmentation. We're able to document along the line. So, looking at this cave fish right here, what do you notice more prominent that wasn't there before? There. You notice that lateral line where all the where all the sensors are. It's coming back. It's looking like the fish that you would catch right out of the stream. And this is what the pigmentation cells look like under high magnification. And that when you really explode them, the pigmentation these are called melanocytes, and they make melanosomes, which are the pigmentation. And all along the body, it makes the dark pigmentation. They also make another type of pigmentation, and that's the yellow pigmentation there. So you can see dark, and then you also see yellow. And you have different cells which make different colors of the pigmentation. And that's what gives birds different plumage colors. You have some that make browns and reds and others. It's the same thing for human hair. On that's why people have red hair. They're making different types of this melanin. And the melanin has multiple functions. You all know we get darker skin to protect ourselves from exposure to sunlight. But melanin protects you from other things that also is a way to bind up toxins in your system and for you able to excrete them. So now, what is the next thing we're doing research on? It's the eye. How do you go from that sighted fish to the blind fish? What is the mechanisms for taking that all off? And in order to study that, you have to study these fish going from a single cell all the way through development. So these are pictures that we have taken at ICR going from a single cell right after fertilization through the initial cell divisions all the way up to these tiny little embryos that are going to develop into fish. And we're able to start to see what is happening and what is changing with the eye. Now, what's interesting about these cave fish is right after fertilization and as they begin to develop, I'll show you the picture on the lower left here, they begin to develop a normal eye. They develop a normal eye right at the very beginning. Within 24 hours after fertilization, they begin to grow an eye. But as you can see in the lower right-hand side, it's just a black dot. Right after almost after 24 hours, the eye begins to degenerate and regress, and it condenses into just a black hyperpigmented dot, and eventually that's completely reabsorbed and blindness is gone. So it doesn't sound like it's a broken system because they are able to develop an eye, but it's a programmed system to purposely shut down eyes after fertilization. And we've been able to take in the generation of the two fish which were raised in the sunlight in surface conditions and map their eye, and they begin to develop a normal eye, which would normally regress or degenerate after 24 hours, but they are keeping their eye for up to 2 to 3 days before it begins to degenerate. So there is something changing in these fish already, but we just don't know the mechanisms for that. So as we end our time, and as you think about this eye, you get that little fish eye in your mind, please pray for us. This is not a money thing. It's not like, oh man, I'll just give ICR more money, help them with their research on that. Well, if you want to give us more money, I'll take it. But It's not money. Money can't buy insights. And the Lord needs to give the insights. He needs to open the things. Oh, this is how it's working. These are the steps. This is what's there. And so pray for that. Pray for that. And I finish up here with this I, so that you remember just what our needs are. Be asking the Lord, help those scientists at ICR, help them to figure out how this is working. Help them to see stuff that nobody's seen before. Help them to understand it like nobody's understood it. Help them to explain it. That's what we really need. So thank you very much. I hope this was educational. You got to see something about these two things, and you now understand two competing explanations for adaptation. Pastor, thank you.